So I'll just repeat what I said in the beginning. I want to thank Ariel Amir for coming to give this seminar. He did a lot of things that I talked about in the course, and he will tell you even newer things than what I told about. So thank you, Ariel.
Thank you very much. It's an honor and a pleasure to be here, and thank you for the beautiful lecture. I could understand most of it, even though I don't speak French, which attests to the physics being a universal language. So let me start, and I'll talk today about correlations and causation in the context of the bacterial cell cycle. And before I start, as you can see here, this is the logo of the Weizmann Institute, and this is the building where I sit in the physics department. And just to put it in a broader context, this is the distance between us and the northern border of the Gaza Strip. And I know this quite well myself because I was a volunteer in an organization that drove cancer patients from Gaza to hospitals in Israel. And one of the people who pioneered this institution is Vivian Silver. And sadly, she was murdered on October 7th. And I hope that this difficult time in the region will improve and wish for peace as soon as possible. And now switching gears to simpler things and stochastic processes. The topic of this lecture will be to use stochastic models to interrogate biophysical processes within the cell, and in particular cell size and cell cycle related. The use of learning from fluctuations and noise to learn about basic processes in physics is an old concept, and one of the early examples are by these remarkable experiments by Perrin, done here in Paris more than 100 years ago, where this is literally the data. He is observing small particles suspended in a fluid and doing a random walk, and this data, coupled with the theory that Einstein derived a few years before, enabled Perrin to prove that matter is made of atoms and to calculate Avogadro's number. And for this, he was received the Nobel Prize. And in fact, a year ago, when I was visiting Paris, I was teaching virtually a statistical physics course at the Weizmann Institute, and I couldn't resist the opportunity, and I went to the Pantheon, which is a few minutes walk from here, and snapped this photograph of Perrin's grave. And you can see he's right across from, if you've got good eyesight, you can see he's right across from Langevin's grave. So I'm sure they have a lot to talk about. And Langevin will also appear in a couple of slides. Okay, so two of my scientific heroes. In a later example related to biology, Luria, a biologist, teamed with Max Delbruck, a physicist. And they also used the large variability, in this case, in the outcome of a simple biological experiment, to prove what we all know today, especially after COVID, that it's random mutations that drive evolution. And if you're not familiar with this example, go and read the original paper from 1943. It's absolutely readable and really beautiful combination of biology and physics. And if you want more examples, I can refer you to a recent book I published on stochastic processes and their applications. And so the theme is to use stochastic models to obtain insights into cell growth. This is the theme of this lecture. And here is an example of E. coli, a bacteria that we all have in our guts, growing and dividing in a microfluidic chamber. And you can see that the cells maintain a constant diameter. They double in length. And after they double in length, they divide symmetrically into two cells. And this is done by the formation of this constriction ring called the FTSZ ring. And I'll get to this remarkable protein later towards the end of the talk. And it's quite amazing that this ordered behavior emerges for the complex interactions of hundreds of different species of proteins in a noisy environment. And this, it's an open problem to understand how this comes about. And indeed, if you quantify exactly from the video I showed you in the previous slide, if you quantify the size distribution, and I think you've heard about this in last week's talk. So you saw this, and I can go quickly. You can quantify distributions by their coefficient of variation. I remind you, it's a standard deviation divided by the mean. And you find that at cell birth or cell division, the CV is about 10%. Okay, so it's a very narrow distribution. And cells need to work to achieve that. So if we follow a lineage of E. coli cells growing and dividing, and if we assume that cells are trying to just time their divisions, we're going to run into a problem. So let's see why is that. So if we assume the generation time is a stochastic variable, even if they divide every 20 minutes plus minus, let's say, 5 seconds, we're going to get a problem because the cell size at the next generation is half because of the symmetry of division. Cell size at the previous generation times e to the lambda t. Lambda is the growth rate that we just understood is constant. Cells grow exponentially approximately at the single cell level. And so if you take this equation and take its logarithm, you will see that the logarithm of v obeys exactly the equation of a random walk, which means it's going to diverge over time. Okay? So generation times must be correlated across generations in a way that will compensate for this and keep the cell sizes distributions to be at steady state and not diverge. Okay? And to impress on you that E. coli is really a remarkable creature, here is another fact. Let's look at DNA replication. In slow growth conditions, this is the bacterial chromosome. It's circular. And in slow growth, it happens in the way you envision it to happen. You duplicate the chromosomes. Now you have 2 circular chromosomes, and cell division takes place. But in faster growth conditions, when you feed the bacteria with good nutrients, something remarkable happens. And it turns out that the doubling time can be 20 minutes, and it's shorter than the time it takes them to duplicate their DNA, which is 40 minutes. Okay? How is this possible? It sounds like a contradiction. This occurs due to the existence of so-called multiple replication forks. Cells start duplicating DNA already for their grandchildren or great-grandchildren. So in a given cell, you have many of these. The orange things here are the origins of replication, the beginning of the chromosome. And you have many of them in a given cell corresponding to these multiple replication forks. So today we know how to measure them. And here is a measurement at the population level where you see a subpopulation of cells with 4 origins of replication and another subpopulation with 8 origins of replication, presumably before and after initiating a new round of DNA replication. Okay? And that leads us to another question regarding the regulation. How does the cell know to regulate the number of multiple replication forks and ensure that every cell born gets an intact copy of the chromosome, which of course is a prerequisite for its viability? All right, so finally the outline of this talk. In the first part, I'll talk about a coarse-grained model for size control. We've just heard about coarse-grained models and their utility, and this will be not looking into the molecular mechanism, but rather describing it phenomenologically. This will give me the mathematical language for the second part of the talk, which will be about the coupling of DNA replication and division. A priori, it's not obvious that the two should be coupled, but I'll show you that they are and use this framework to analyze experimental data using a framework I will introduce called causal inference. And in the third part of the talk, I'll talk about the molecular mechanisms, very recent work starting to look into the molecular mechanisms involved. Now, usually when I talk to physicists, they only care about the coarse-grained models. Biologists only care about the molecular mechanism. So everybody is going to be equally frustrated with this talk, no matter what their background is. All right. So using correlations to decipher size control, several years ago, we devised this coarse-grained model to try to describe size control phenomenologically. And the idea of this work is really inspired, as I mentioned, by the Langevin equation. Okay, so what is the Langevin equation? dx to dt, this is a simple A Langevin equation. It's more general than that. But one of the simplest one you can write is dx to dt is a function of x. This is, say, the feedback on x, the position, or the force, which is position dependent, plus a stochastic noise. So the key insight is to decouple things into a deterministic component, this is f of x, and a stochastic term. And in the same way, we suggested, let's think about cell division as being a function of the size at birth, This is quote unquote what the cell is trying to achieve. And to that, we're going to add the inevitable noise due to the stochasticity in the noisy environment of the cell. So to give you some examples, if this function f is a constant, it means that cells are trying to get to a critical size and then divide. And for that reason, this is often referred to as a sizer. If cells try to double their size, they're born at size VB, and they want to divide at twice VB, again up to these fluctuations and noise, because growth is exponential, trying to double yourself is equivalent to grow for a constant duration. So this is called a timer. And this is exactly the model we analyzed before, and we showed it doesn't lead to size control. It leads to these diverging distributions. So this is called a timer, and we know it's not going to be a good solution. And if size at, what you're trying to do is add a constant from size at birth, so size at division is size at birth plus a constant, this is called an adder. And I want to emphasize that these are just 3 examples in a continuous spectrum of model. There is nothing really special about these 3 examples, even though they see a lot of use in the literature. And in fact, because of this small CV, the small width of the size distributions, we can tailor expand, and we only care about the behavior of f near its typical value that I call V0. So this is, say, the average size at birth. And if I call the derivative 2 times 1 minus alpha, I now have a continuous knob alpha that takes me from 0 for the timer to a sizer where this is 1 and the derivative is 0. And the adder is precisely in between these two because it has a derivative of 1 and therefore a value of alpha equals to half. Okay? And feel free to interrupt with questions throughout my talk. Okay, so after some simple algebra, one finds that if you work with a logarithm of size, and this is under the assumption of time additive noise, the nature of the noise doesn't matter for all the conclusions I'm going to tell you. Okay, so you can add either noise to the size or noise to the generation time, and you'll reach the same conclusions. And under these assumptions, you find that log of size at the next generation is a constant times log of size plus noise. And this is known as a discrete stochastic map or an autoregressive process. And for alpha equals 0, this is just the equation of a random walk, like we saw before. So you'll see that log of size will do fluctuations and will diverge. But if you take, for example, alpha equals to half and simulate this on a computer, which is very, very easy to do, you get fluctuations around the steady state value that seem to mimic what you get from experimental data. This is data tracking E. coli cells in microfluidic devices over many, many generations. And one can quantify that. And I want to also point out an analogy with, again, those coming from physics. One of the simplest Langevin equations we can write corresponds to the so-called Ornstein-Uhlenbeck process, corresponding to a particle in a harmonic potential, confining potential. So then this harmonic confining potential gives rise to a linear restoring force. Without this restoring force, this will be, x will run away to infinity due to this noise driving it, the random walker. But in the presence of this restoring force, we will ultimately get a stationary distribution that in this case is Gaussian. That's the Boltzmann distribution for this harmonic confining potential. And something similar happens here for our discrete stochastic maps. But in this case, you get a stable size distribution only when alpha is in the range between 0 and 2. So 0 is out because of what we saw before. But say both adder and sizer lead to steady state, stationary steady state distributions. And what I'll show you in the next couple of slides is that for some reason that we still don't know why, adders were chosen by nature and are very prevalent across the tree of life. Okay, so what is the strategy? Using this model and simple calculations allows one to calculate correlations and distributions between important cell cycle variables. So for example, I can look at cell size at birth and cell size at birth, say, 2 generations apart, and calculate the correlation between these 2 variables. And when you do that, and maybe you did that last week, you find that the correlation decays as 1 minus alpha to the power of n, where n is the separation in number of generations. And similarly, if you calculate the correlation between size at birth and the generation time, you find a negative correlation. Why negative? Because you need to compensate. If you're born small, you want to grow longer to compensate for that cell size. And it turns out to be minus root of alpha over 2. And now we can follow the same philosophy I mentioned earlier in the talk, where we want to use the natural variability between cells as an alternative microscope to probe these processes. And when we look at data, one finds that a cell and its daughter cell have a correlation of about 1/2. A cell and its granddaughter cell have a correlation of about 1/4. Both of these suggest this value of alpha equals to 1/2, according to this formula. And indeed, that would predict a correlation of -1/2 between cell size at birth and its generation time, which is exactly what was reported in the literature work by Lydia Robert, also here in Paris. And so this suggests to us this alpha equals to 1/2 model corresponding to this adder. And I want to give you a different way. Sure. Yes. Okay, so correlations, coefficients, these are Pearson correlation coefficients. So they are the expectation value of x minus x average, y minus y average, normalized by their standard deviations, by the product of the standard deviation. So they go between -1 and 1. A variable in itself is perfectly correlated, it's 1. A variable in minus itself is -1. And in this case, you can see after a calculation that within this model, the correlations between size and generation time have to be negative in the viable stability range. In the range alpha between 0 and 2, these correlations are always negative. Because if a cell is born, okay, great question. So if a cell is born, it doesn't have to be positive. It doesn't have to be positive. It's 1 minus alpha. In general, say a cell and its daughter cell, it will give you 1 minus alpha. Alpha is in the range 0 to 2. Okay, so it can be, if it's 1.5, it'll still give you stable size distributions, but this correlation will be negative. It's overcompensation. When alpha is larger than 2, you've overcompensated too much, and you become again unstable. But the first one doesn't have to be positive, but we find experimentally, and the first paper is actually from 1980, and they reported a value of 1/2. So it happens to be that these special values that correspond to alpha equals to 1/2 are found for E. coli, but certainly it could have been otherwise. Other questions? Okay, so I want to give you a different way to understand these adders or to infer these adders from experimental data. So what I'm showing you here, every circle is a single cell. So we're learning again from fluctuations of single cell. X-axis is size at birth, y-axis is size at division. And this is a heat map, so most cells reside here in this region. And if biology was perfect, we will just have a very bright dot here, and we wouldn't be able to learn anything. So really, we need the variability in order to do this analysis. Now we can do a procedure called binning. What is binning? We take this rectangle and basically condition on the size at birth and average over all the cells in this rectangle, and that gives us this red circle here. Okay? No interpretation, it's just averaging over the raw data. And when we do this, we get these set of red circles. Again, no interpretation, but they happen to have a slope of 1 corresponding to the equation VD equals VB plus a constant. So cells are essentially adding a constant plus a noise that gives rise to this cloud of points. And so that's another way to arrive at the adder correlation using the procedure of binning. And I also want to mention, I'm going to refute this model later in the talk, but it's good to have in your head. So how do I think about these adder correlations? The simplest way to think about the adder model, how do you add a constant from birth to division? Imagine some protein that is being accumulated from birth, and it reaches a critical value to trigger division. So this is called the threshold accumulation model, and would accomplish an adder, at least in principle. Okay, I want to now show you something that surely you haven't seen. Oh, I want to say one more thing. Within the same model for an adder, the model makes a prediction that if you plot log to the base 2 of size normalized by average size, it would collapse with a normalized time, doubling time divided by average doubling time. without any fitting parameters, and this is what you find. This is not simulation, this is actual data on E. coli, and you see this beautiful scaling collapse. And I want to show you another result suggesting that this adder model can capture the statistics well for E. coli, and this is unpublished work. And there is actually a nice paper that I often recommend my students called Writing Physics by David Mermin, a renowned condensed matter physicist, where he says that basically the process of writing a paper is part of the scientific discovery process itself. So I often recommend my students to start writing earlier, even before they finished all the results, start writing the paper and you'll learn and you'll see where the gaps are. And I think there is something similar about presenting physics. And actually when I presented, I prepared some slides for an ERC defense a few months ago, and one of the people who gave me feedback said, what you're saying makes no sense, because when I look at these heat maps, this is the same data I showed you in the previous slide, you can see that the contours don't align with this line with slope of 1. So the binning, whatever, it's the binning, but the contours don't align, and what you say makes no sense. And together with the work of Partita Carr, a PhD student in my group, we did the calculation. You can all do this calculation after what you've learned last week. It's not a complicated calculation, but the result was very surprising to me. When you calculate the contours, you find that they're actually tilted, and they sit at an angle within the adder model. You predict them to sit at an angles of arctangents 3 plus square root of 13 over 2, which is about 73 degrees. And that's what you see in the data. So actually, the discrepancy between the binning and the contours is expected within the adder models, and is another evidence that the model can capture the statistics of the actual data pretty well. So this is unpublished results. Okay, so we saw so far that this adder model can capture the statistics well on E. coli. And then there is a famous quote by Jacques Monod. We heard about some other works he did, but he has this famous quote, what is true for E. coli is true for the elephant, which I don't expect you to take too seriously in this context. However, in work with collaborators, we've shown that budding yeast, the eukaryote, also shows the same adder correlation. Now you understand this is the bin data, and it has a slope of 1. And also in archaea, called H. salinarum, an extremophile that grows in very high salinity concentrations, such as the Dead Sea in Israel. And if you ever use the Tibetan sea salt, this is the pink. This is what gives the pink color to your Tibetan sea salt. And these very different organisms all showed these adder correlations. And by now, by work in other groups, showed that adders are observed for all 3 domains of life, including human cancer cells and 8 bacterial species. And this is how it looks like on the phylogenetic tree of life. And I should also point out this is a nice work on showing near adder behavior by certain mammalian cell lines, work by Matthew Peel and collaborators here in Paris. I think he's also going to talk he spoke already, okay, so another connection. And this led us to ask the question, are adders optimizing the population growth? You see that they evolved multiple times across the tree of life. Is there anything optimal about these adder control strategy? And we thought that maybe they're optimizing the population growth. So again, if we look at this lineage tree for E. coli, the number of cells grows exponentially. Maybe they're optimizing this exponent here. And we've spent a lot of time thinking about this. And within all the models we considered so far, the size control strategy did not affect the population growth. I'm not going to talk about it today. If you're interested, this is a review we wrote on this a couple of years ago. So as far as I know, this is still an open question why adders have evolved, why they're so prevalent across the tree of life. Okay, so far I discussed a coarse-grained model for size control, and I showed you that a simple model where you add a constant volume from birth to division explains the statistics very well. And DNA replication was not involved in any way. But now I want to switch to the second part and talk about how DNA replication and divisions are coupled and convince you that they are. And so a couple of years ago, work by a former PhD student, Po-Yi Ho, who recently started as faculty at Westlake University in China, we showed that if you look at a different class of models, all the kind of the Langevin type model I showed you was centered around birth. Cell size at division was assumed from the very start to be a function of cell birth. But what if we switch things around and put DNA replication at the center? So instead of division being controlled by birth, maybe division is downstream of DNA replication initiation, the point in time in which I start replicating another round of DNA, and we control DNA replication instead. And we showed that if you keep this adder, but not from birth to division, but from initiation of DNA replication to the next initiation event, so I still have an adder, but not centered around division, but centered around DNA, then all the correlations and distributions I showed you are inherited from this model. They remain intact, but you can explain additional observation that I'm not getting into because of lack of time today, but it's known in E. coli that the initiation volume, the size at which, on average, the size at which you start initiating DNA replication is independent of the growth rate. This is called the constant initiation mass. And it's intimately related to another growth law. We heard a lot about growth laws in the previous hour. And there is a famous growth law actually from the 1950s saying that the size of E. coli depends exponentially on its growth rate. If you give E. coli better food, they're bigger on average. And this can be naturally explained within this model. And further, it automatically regulates the number of multiple replication forks, as I introduced early on in the talk. So now we have two classes of competing models, okay? And I'm kind of zooming out again now. In the first part of the talk, I spoke about division as being completely uncoupled from DNA replication, as in the model where you're adding the volume from birth to division, or variants of this, like this one, add a surface area from birth to division. So this is a class of models where these two processes are uncoupled. In a second class that actually dates to the 1960s, DNA replication triggers division. Okay, so division is downstream of DNA replication. And in another more recent variant, due to work from Marco Costantino Lagomarsina, that again you're going to hear from in a couple of weeks, it's a kind of hybrid of these two. This is called the concurrent processes model. And it says in order for division to happen, we need two conditions to be obeyed. One of the conditions is integrating these signals from birth, such as the threshold accumulation of a protein from birth. And the second is related to DNA replication. And you need both cues to arrive in order for you to go through division. And what I'm showing you below here are graphs representing these different class of models. For example, here birth and divisions are coupled, and Li here stands for initiation of DNA replication, Li minus 1 and Li are the DNA replication cycles, and they're uncoupled from each other. In the opposite limit, the initiation of DNA replication triggers division, and this is the concurrent processes model, where for division to occur, you have cues from birth as well as cues from DNA replication. Now, this is not just a graphical representation. This has a very precise mathematical meaning, and every arrow in this graph corresponds exactly to a discrete stochastic map like I introduced in the first part of the talk. So we invented this for cell size control, but it turns out that this language and this graphical representation is well developed and is known as causal inference and pioneered by Judea Pearl and others. Okay, so I'm going to give you now some examples of what causal inference is, some warm-up examples, and then I'll mention the kind of the theorems that people know in this context. Okay? So I'm going to switch gears to something different in order to illustrate the concept of causal inference. And here is an example. So I took it from this paper from 2016. It turns out there is a positive correlation between coffee drinking and lung cancer. And for somebody like me that likes his coffee, this is very disturbing. But luckily, in this paper, they propose another model. Perhaps smoking is causally linked to coffee drinking, and of course, smoking is causally linked to lung cancer, and that's why these two variables are correlated. Now, in both the top model and the bottom model, A and B, if we think about them as random variables, are going to be positively correlated. So how can we tease apart these two models? And the trick is to look at what's called conditional independence tests. So the idea is, let's look at the correlation between A and B when I condition on the variable C. And if the bottom model is correct, if I only take the subset of people that say smoke, now the correlation between A and B should vanish. Okay? So that's the idea of conditioning on a variable in order to test models. So this makes a testable predictions we can now use to distinguish between these two models. And we can generalize this. And again, this was pioneered by Judea Pearl. And we can look at what happens to x and z when we condition on a variable y. And so, for example, if you have a different model where x leads to y leads to z, x and z are going to be correlated. But if we condition on y, we make them uncorrelated. And this is called closing the path between them. In another example, if x and z affect y, x and z are by assumption uncorrelated, but now when we condition on y, we actually make them correlated. So that opens the path between x and z. And this is the example we just did, where a variable y affected a variable x and z. X and z are correlated, but when you condition on y, we make them uncorrelated and close the path. And so there is a well-oiled machinery, and I'm not going to go into the details here. They're not important, I think, for understanding the rest of the talk. But if you give me a general directed acyclic graph, and you tell me you're conditioning on this and this and this node, the theorem enables you to know whether two other variables are going to be correlated or not. Okay? And it's a very powerful machinery to distinguish between models. And so what I'm going to do next is use data that I will explain to test which of these three classes of cell cycle models are consistent with the experimental data. Okay, so the data we're using is a wonderful collaboration with a group of Jan Manik at the University of Tennessee. And what they do is use fluorescent reporters that you've also heard about in this course. And they use fluorescent reporters that mark the replication fork. So you can see a replication fork started here and terminated here. And using this and microfluidics, they can get the key cell cycle variables, including cell birth, size, and time, the size and time at initiation and termination of DNA replication. And using another reporter, FTSN, they can find when the cell starts constricting this Z-ring that I mentioned in one of the earlier slides, when the cell starts pinching into two daughter cells. And so they can get all of these variables for E. coli at 8 different growth conditions for hundreds of cells. And these conditions range from fast to slow, where by fast and slow, of course, I mean, are there multiple replication forks or not? This is the qualitative difference between what I mean between fast and slow. And I will not discuss, this collaboration started just before COVID hit, where we met at a conference and teamed up. And I will not discuss two other works that came from this collaboration, but if you're interested in this topic, then I refer you to these two publications. One more thing I should say. When we work with this real data, conditioning is not the naive conditioning like in the case of smoking, right? Smoking, we have smokers, non-smokers. I just dilute my population size by 2. I can deal with it. If I condition on, say, cell size being 2.2 microns at birth, I will go from hundreds of cells to maybe 5 cells, and I can't afford that. So there is a trick to be able to use the entire data set and still condition. And it's kind of a technicality, so feel free to ignore this slide. But the trick is we define new variables called the residuals, which remove the correlation with the variable we're conditioning on. So say, instead of the variable A, I take A minus its correlation with the parent variable C multiplied by the value of C. And you can see that, say, for this example, even though A and B are correlated, the residuals, A tilde and B tilde, become uncorrelated. So this is a way to condition while maintaining the complete size of the data set we're working with, and that allows us to condition even on multiple variables, if we like, without reducing all the time the size of the data set. Okay, so now let's go through the examples. So the first thing we did was look at this constriction event that is often ignored. People, when you heard about cells birth and division, people ignore the fact that actually before you divide and the cell becomes 2 cells, you actually need to constrict. So we said, how does it work? Is constriction commitment to division? And now you know what this graph means. This graph means that cell size at division is a function of cell size at constriction plus some noise and so on. So is this the right graph? Or maybe once you start constriction, in order to divide, there are still additional cues like this protein accumulation from birth that needs to be met in order for division to happen. Okay? How do we tell these models apart? In both of these models, all three variables are all positively correlated for the adder. Okay? But if this model is correct, what do we need to do? We need to condition on the middle variable. So if we condition on size at constriction, the adder correlation between birth and division will vanish. We'll get zero correlation. By the way, I should mention, I just mentioned adder correlations, but the whole beauty of this causal inference formalism is that it's actually completely robust to the details. I don't need to fit precise value to the correlations. All I'm asking is the correlation zero or non-zero? And in that sense, it's a very powerful and robust method to use. So when we apply this to the data, we find that when we condition on the size at constriction, the adder correlation with the slope of 1 vanish completely. We get essentially flat lines, suggesting that the simple picture is the correct one in this case. Okay, so constriction is really a checkpoint in the sense that once I go through constriction, I commit to division, and division doesn't care about anything that happened before constriction. Okay, so now we're going to the more complex question of control over division, or rather over constriction, because we just realized that we can go one step before and ask what happens at constriction. And these are now the causal inference diagrams we're looking into. One is that the cycles are independent. Replication, DNA replication is independent of cell division. This is the one where initiation of DNA replication controls constriction, which in turn, as we just saw, controls division. And this is the framework corresponding to the concurrent processes picture, where both cues from initiation of DNA replication and from cell birth are important for determining the point at which you go through constriction. And first of all, in these two cases, LB and LI and LC are all correlated with each other, while here we expect, because these are independent, then LB should be uncorrelated with Li. So in order to distinguish the right model from the left two models, we don't need causal inference. We can just look at simple two-variable correlations. We don't need to go into these fancy conditioning tricks. And when you do that, you find in all growth conditions that the initiation size is correlated with the size at birth. Similarly, the constriction size is correlated with the initiation size, and that rules out this picture where these two processes proceed independently of each other. Okay, so again, this is not causal inference. This is just your plain vanilla correlations, which are also a good tool to use whenever you can. But now we need to use the other machinery, because now that all these variables are correlated with each other, how can we tell apart this model from this model? And you can kind of guess the answer. We're going to condition on this intermediate variable Li and see whether we've washed away the correlations, say, between LB and LD. Okay? So this is what we do. We look at, in this case, at the correlation between LC and LB. You can see that the path, if the left model is correct, the only path from LB to LC is this one. Okay? And if I condition on Li, I'm going to wash these correlations and destroy these correlations. What do we find? Unfortunately, we find something more complex. We find that in slow growth conditions, okay, so this is the regime where there are no multiple replication forks, you get a flat line, suggesting that in slow growth conditions, DNA replication controls cell division. So it's kind of the simple version, opposite of the version where birth controls division. No, it's DNA replication that controls division. However, in fast growth conditions, we find that this is a non-zero correlation supporting the concurrent processes model. Okay? So it's a more nuanced answer that the replication controls division in slow growth, but we have simultaneous control of both replication and birth-related processes in fast growth conditions. And this is actually very resonant with a paper we published in 2022, where we reached the same conclusion in completely different methods using molecular perturbations and mutants. So it's always nice to reach the same conclusions in very different ways, and also shows that this phenomenological coarse-grained modeling is not orthogonal to the molecular perturbations, they can often work synergistically together, and I'll get to that point towards the end of the talk as well. What about control of the DNA replication cycles? Okay, so putting aside division, the picture that was accepted in the literature was this adder per origin model that we proposed back in 2015, saying that the control of DNA replication, the size, you add a constant volume from initiation of DNA replication to the next initiation. This is this adder per origin where you're adding this volume between 2 rounds of DNA replication. And this is the diagram corresponding to it. Of course, initiation of DNA replication leads to termination of DNA replication. This is just the process starting and going, and we know all about it using, we know the speed at which these replication forks move using single molecule experiments. So this is well characterized. And this is the picture we expect. And if this is correct, then we expect an adder between size at initiation and size at the next initiation. This was observed. I'm not showing you the data, but this was seen. However, using causal inference, this would tell us that if I take the size at initiation in the next round and condition on the size at initiation in the previous round, I should get no correlation with the size at termination. And this is not what we found. So both in slow growth and in fast growth, we find positive correlation, and that suggests to us that we're missing an arrow. And there is potentially another link between termination and initiation in the next round. And so the control of DNA replication forks is probably more complex than what we thought earlier. And again, this is resonant with work from, in this case, Johan Elf's group, coming from very different direction using mutants and molecular manipulations. Okay, so I think there is a lot we don't understand about how these things are regulated. And in the remaining couple of minutes, this is a graphical summary of everything I told you in the second part of the talk. So this is kind of the picture we have. The replication cycle is upstream anyway. It doesn't care about division. So initiation and termination are coupled and lead to initiation in the next round with, again, this potential link from termination to initiation. So this is the replication of DNA. In fast growth conditions, we have an additional link from birth determining constriction, while in slow growth, only DNA replication matters for the onset of constriction, and that, as I showed you, leads to division. So this is kind of summarizing everything we've learned so far from using these causal inference tricks, which I think, again, I should emphasize, this is applicable for anybody working with data. It's good to keep in mind this causal inference framework because it's a good tool to have that I think physicists are quite ignorant of. All right, so now in the remaining few minutes, I want to talk about molecular mechanisms. And this is a work from very recent, work that was submitted about a week ago. And we're starting to ask, what are the molecular players implementing these strategies? And one key candidate that has been around in the literature is FTSZ. So FTSZ is this remarkable protein that, as the cell cycle progresses, forms this so-called Z-ring that ultimately divides the cell into 2. And a lot is known about it. It does treadmilling, and I will not get into it. It's less important for the purpose of this talk. It doesn't work by itself, of course, so there are tens of different proteins arranged in this complex around the Z-ring that together need to coordinate the synthesis of cell wall to divide one E. coli cell into 2 daughter cells. So it's a highly complex process involving many players. And it was proposed that maybe the other correlations come from the accumulation of FTSZ from cell birth to division. So this is kind of the simplest picture. A cell is born, it accumulates enough FTSZ until when you have enough of it, the cell divides. And we've set out to test this picture using molecular perturbation. And as I mentioned, you can find it on the bioRxiv. It's from March 1st. And what Jan and his team did was upregulate FTSZ in a very precise fashion. So you have E. coli growing in microfluidic channels, and you're able to upregulate FTSZ in a way that you can quantify and fits a simple model extremely well. And what happens? So as you expect, if FTSZ is a protein that is required for constriction, if you upregulate it, constriction should happen earlier, and you expect to get shorter cells. And this is exactly what you do. So the dashed line is the time at which you upregulate this protein in a controlled manner. And you see that this is average over many cells, and you can see that the length decreased in a stepwise manner, more or less. And furthermore, what happened to the doubling time of the cell? You see that initially, you've just suddenly you're ramping up the level of FTSZ, cells become shorter, and they should divide quicker. So initially, you get a transient dip in the doubling time, but ultimately, the doubling time should go back to exactly what it was before. Why? Because just like we heard, doubling time is set by completely different things, ribosomes, and it's all about growth rate. We're not changing the growth rate by upregulating FTSZ. So ultimately, the doubling time must recover to its original value. How would that come about mechanistically? Well, I have a higher FTSZ concentration, but I have smaller cells. And the total amount of FTSZ I'm going to make is exactly the same. So I must transient to the original doubling time. So qualitatively, this seems to be consistent with FTSZ being the limiting resource for cell constriction. However, we find that that doesn't work quantitatively. And if you think about this simple threshold accumulation model, doubling the concentration of FTSZ should give you cells that are exactly half in size. Okay, so the calculation is one line. There is nothing to it. And this corresponds to this dashed line here. And that doesn't quantitatively agree with the data. The different colors here correspond to different levels of upregulation. And what you see is that this line agrees much better with the data, and that's precisely this concurrent processes model, suggesting that FTSZ is an important player, but it's not the only one. And to determine constrictions, you do need to accumulate enough FTSZ, but there are also additional cues that we believe are related to DNA replication, like I showed in the second part of the talk. And within this concurrent processes model, you can quantitatively explain the shortening of the cell as well as the transient decrease in the doubling time of the cell. Okay, so again, I want to emphasize, here we've used molecular toolbox of genetics, but we've reached the same conclusions we've reached before using causal inference, namely that in this condition, concurrent processes model does a good job of explaining the data. All right, I'm finishing my talk, but I want to kind of zoom out for a second and ask, what about cell density? You saw that in this threshold accumulation model, having a constant density of the molecular players is key. And this is a different story that I didn't touch in this talk. We know that cells constantly pump ions and other molecules into and out of the cell, and transport played a role also in the previous modeling, the previous lecture. And this affects cell volume and density through water flows. This affects the turgor pressure, the membrane voltage, and it's absolutely critical for physiology of every cell, including E. coli cell. And I think that the role of electrophysiology, in particular in the context of bacteria, is a really fertile ground to connect to what I discussed today. And I don't think it's orthogonal axis. I think these things should be studied together. And I don't know if you're going to study it in this course, but there is beautiful works about how cell density is maintained. Both of these papers focus on eukaryotic cells. But I think the principles should be applicable to E. coli as well, potentially with some modifications, because E. coli has a huge turgor pressure of about 1 atmosphere, 1 excess atmosphere, which is remarkable. And so I think this is something that I hope will be understood better in the future. And with that, I'd like to summarize. So I showed you that correlations can be used to construct coarse-grained models for size control. I showed you that others are prevalent across the tree of life for reasons that we don't know. I showed you that these causal inference and conditional independent tests can help narrow down the viable models. By the way, they don't tell you what the model is. It's not a black box where you put in the data and it says, here is the model. It's good in the cases where you have several models that are the candidates. And in some cases, like the one I showed you today, you can rule out certain viable models using this approach. Okay, so it's not a magic thing that you put in data and you get the model. This analysis allowed us to support models where DNA replication is coupled to division. In slow growth, DNA replication triggers constriction, and in fast growth, you have additional cues such as the accumulation of FTSZ that I showed in the last part of the talk. And molecular mechanisms in general are synergistic to coarse-grained modeling and can strengthen the conclusions. And I'd like to thank my funding sources, and I'd like to thank my current and former students. In particular, Pratita Kar was absolutely instrumental in driving this whole causal inference analysis over the last couple of years. And the collaborators over the years, and in particular Jan and Jana Manik from the University of Tennessee, that were close collaborators on the causal inference project as well. Thank you for your attention.